So thanks for that wonderful introduction. Wait for the slides. These are my disclosures. It's been great to be here. I've been thinking about how this is an international workshop, the themes of international. And as I was going for my run to wake up yesterday, on this beautiful, beautiful day that was yesterday, I found all these kids and youths on the meadows with protesting and vouching for climate change. And I was thinking about this because only knowing that hours later, my two daughters would actually be also in Boston Common doing the same. So it was kind of interesting to think about common shared thoughts and ideas, but back to CLL. So we just heard from Gadi all the work that has been done by us and collectively by many people in this audience about identifying drivers in CLL. We understand very well the genetic heterogeneity that's present in CLL. Shown here are just in the, let's see, whoops. Actually, I'll move on, but I think by now we know very well that there's many, many different drivers that have been identified. And so the questions that my group has been asking is, okay, we've identified all these drivers, they're putative drivers. What actually is the functional impact of these mutations? And can we understand that? And furthermore, can we start to integrate the layers of genetics and expression to understand what are the features important for therapeutic resistance? So I'm going to share some new and also unpublished work. And really the idea that we had was, well, okay, if we understand there's all this genetic heterogeneity in our patients, can we, for example, generate faithful mouse models? We know how hard it is to manipulate using our suite of conventional genetic tools, patient samples and B cells, but maybe in a well-trodden path, can we also think about mouse models? And in doing so, can we then dissect mechanisms of disease progression and transformation? And this will also let us test novel therapies. And so we already started on this path a number of years ago. You already heard earlier today from Lili Wang that we've had a long program, as well as from Gadi that we've had a long program in thinking about mutated SF3B1. Lili already, a number of years ago, performed a deep transcriptional characterization of SF3B1 in human samples, and our more recent work has been to try to model that in mice. This cover on Cancer Cell was kind of inspired by a Chinese proverb, which is this idea that a dragon doesn't come to life until you finish dotting the pupils with the paintbrush. And so what we see here is we have already one paintbrush with the SF3B1 that is there, but the dragon within doesn't come through. The CLL isn't released until we also have cooperation together with deleted ATM. And so this idea that SF3B1 requires cooperation in order to generate CLL, and certainly Lili and Sangye Ying in the lab identified that these senescent B cells are then become fully functional with the deleted ATM to generate CLL. So having gone on that path and seen all the what we could learn from this mouse model, we started to think, well, maybe we should start to model some other mutations. And we were drawn to IKZF3 because, as Gadi mentioned, we discovered new drivers. This is one of the drivers that we identified in our cohort of 538 samples. And what interested us in IKZF3 is that it's actually a known transcription factor that's important for B cell development. And what we identified in that original cohort is that there was actually a hotspot mutation in a DNA binding domain. So certainly interesting, suggesting some sort of gain of function perhaps. And furthermore, in looking at a cohort, the CLL8 cohort together with the German CLL study group, we could see that there was an increase in CCF of IKZF3 in the setting of resistance to chemoimmunotherapy. And so we went along the strategy of creating a mouse line to model mutated IKZF3 in a heterozygous fashion that would be expressed in the presence of CRE. So you can see we took the mice we actually bred it together with CD19 Cre. So now we have a mutation that has expression that's limited to B cells, and we could generate and confirm that we could generate cohorts of wild type, het, and homozygous mice. And so our question was, if this was going to be a transcription factor affecting B cells, can we actually see some functional effects? Can we look at B cell differentiation using immunophenotyping? Do we see differences in the subpopulations of B cells? And it was quite clear that there, in the mutated mice, het or homozygous, we certainly see decrement in the marginal zone B cells, proportional increase in the follicular, and we could see that both by flow and also by immunohistochemistry. And this was quite different than what we had already noted in the SF3B1 mice, where we saw actually an increase in the marginal zone. In terms of antigen-specific immune responses, we used a very conventional approach, which is sheep red blood cell immunization. And what we could see with the IKZF3 mutated mice was increase in germinal center formation following immunization. And this was also quite different than the SF3B1 mice, where with sheep red blood cell immunization, we actually saw relative decrease in the number and size of germinal centers, actually the size of germinal centers compared to the wild type. So these type of changes in the young mice were actually associated with transcriptional changes, and what we could see is that differentially there was actually impact on BCR signaling, which we could confirm through Western blotting, so increased phospho-Syk, increased phospho-AKT and ERK, and also impact on chemotaxis. And again, this was quite different than what we saw in the mutated SF3B1 case. And finally, as we started to age these mice out a little longer, we could actually see that IKZF3 alone, the heterozygous mice, appeared to generate CLL with some frequency. So by now, this cohort is still aging, but we do see that 23% of the mice at a certain age develop CLL. There is some degree of lymphoma as well. Different than mutated SF3B1, we actually do not see involvement of the bone marrow, so already some phenotypic differences. We do see involvement in the spleen and the peripheral blood. Interestingly enough, for the homozygous, we see early death. We still don't know why, and this is under active investigation. So just to finish this part of the talk, I just wanted to describe our new work looking at mutated IKZF3, which is already showing us that there's a unique transcriptional signature associated with BCR signaling, activation. There's marked changes in signaling and migratory capacity. It can induce CLL in mice alone by itself, so this was quite interesting to us. I mentioned before that SF3B1 required cooperation with another driver in order to see CLL, so very different biology. And so as we go on our path to trying to develop models that are reflective of the heterogeneity that we see in our patients, I think that this is an illustration of maybe at least two different poles that we're seeing. Now I'm going to pivot to a second work that we've been thinking about, which is the role of genomic changes in resistance. And a longstanding theme of work that we've done in our group collaboratively with Gaddy's group is to examine clonal evolution, the selection of fitter clones, and already for some time now, because of our interest in defining this intratumoral heterogeneity, we've known that this type of complex mix can set the stage for subsequent evolution and resistance. And we just heard from Gaddy the work that we've done in the natural progression setting. And we've also been very interested in looking at the impact of therapy. We've already reported out our work in chemotherapy, targeted therapy, and more recently, we've been working in the immunotherapy setting. And so I think that as we start to move forward into where we are today, we have venetoclax. That's for this audience, it's a super interesting drug, and I don't think I need to go through the mechanism of this BCL2 signaling inhibitor, but our question is, can we identify mechanisms of resistance now to BCL2 inhibition in lymphoid malignancies? And I point to new work that just was reported out a few days ago online at Cancer Cell, led by Roman Guiez, who's in the audience, who was a postdoc in my lab, currently in Clermont-Ferrand, and together with Vivian Liu, extraordinary Harvard medical student, now an intern at University of Washington. So we kind of, for this work, we went upon a path that we had taken before, which is to collect samples from patients before and after, at the time of resistance to venetoclax. Altogether, we examined 6 patients. This was done together with Jennifer Brown, Matt Davids, also working with Phil Thompson and Bill Weirda at MD Anderson. And what we did was perform whole exome sequencing using the tools that Gadi had already mentioned. And when we looked at these 2D plots where the x-axis is the pre-treatment cancer cell fraction, the y-axis is the post-treatment cancer cell fraction, we clearly see that there is clonal shifts going on. We did not find any BCL2 mutations. We didn't see any consistent CLL driver that was selected for resistance, although we did see this interesting pattern in patient 1 and 2 where both of them had clonal SF3B1 and TP53, and both had a rising subclone that had deletion 17p, del 8p, and amplification of 8q. So interesting, but certainly nothing kind of unifying that we could kind of put our head around. And so we just developed a second approach, which is we thought, okay, well, why don't we come and think about a genome scale screen and systematically try to identify determinants of resistance against venetoclax. And for this, we use the OCI-LY1 lymphoma cell line, performing both in parallel loss of function screens by adding a CRISPR library, and also a gain of function screens, so overexpression of tens of thousands of ORFs. And in doing so, the results were really quite striking. So first, in terms of the CRISPR screen, we happily identified those targets of the BCL signaling pathway as we would expect, but also identified a number of different transcription factors shown here that have already been previously shown to be mutated across other B-cell malignancies. So certainly interesting, and I point your attention specifically to ID3, who's going to come up a little bit later. The overexpression screen showed a different set of results, which was also interesting. So again, we found targets in relationship to BCL2 signaling in the right directionality, but we also found multiple regulators of the AMP signaling network. So both on the C-AMP the protein kinase A pathway and the related AMPK pathway. So to start to help sort all of this out, we also developed a resistant cell line through chronic exposure to venetoclax, and this we used to help try to figure out signatures by transcriptome or proteome and perform some functional investigations. And so as we started to characterize the cell line, we began to see, for the most part, there was some degree of concordance between the RNA-seq on the x-axis, the proteome on the y-axis, the signals. But it turns out that the intersecting these results together with our screen results, we could see that the one consistent signal was not unexpectedly MCL1 overexpression. We certainly see by Western blot high expression of MCL1 in the resistant cell line. And we see also evidence of synergy between venetoclax and an MCL1 inhibitor in the treatment of this cell line. Now, we also unexpectedly saw many different determinants related to cellular energy metabolism. So that's shown by the pink triangles. And this was interesting because there was already work reported that lymphoid transcription factors are metabolic gatekeepers, so something to keep in mind. And we know that also AMP-K, which had kind of shown up on the overexpression screen, is a key regulator of cell metabolism in OXPHOS. And so trying to kind of link these different sort of ideas together. And so when we think about OXPHOS, we know that that involves the inner membrane of the mitochondria. We know that there are specific inhibitors that can help us figure out impact on the electron transport chain. And together with the Wamsi Mutha and Alexis Jordan at the Broad Institute, we performed seahorse assays on the OCY LY1 cell line as well as multiple other resistant cell lines. And we could see consistently that the resistant lines appeared to have increased basal and maximal mitochondrial respiration, so kind of fitter mitochondria. And as we started to go back and look at our cell lines, we could see that using these electron transport chain inhibitors that it did render the sensitive lines. There was synergy that we could see in the sensitive lines and increased sensitivity to venetoclax in combination when we looked at the resistant line. And this we could also see in an in vivo model where the combination of oligomycin and venetoclax actually had an impact in tumor volume. So with all this, can we confirm these findings in our patient samples? So this was all sorts of cell line characterization. Before getting there, we actually tried to also integrate some genetics as well. So we looked at the resistant versus sensitive cells. Daniel Rosebrock in Gaddy's group identified that there was an AMP1 chromosome 1 amplification. And when we looked at the cell line, and then we also looked at the 6 patients that I talked to you about in the very beginning, it turns out that 3 out of the 6 patients actually also had amplifications within the same region. And when we look in that region and see what's within that region that's shared, we actually see MCL1, and we also see another regulator of the AMP-K signaling pathway. And so fortunately, we had blocks tissue from these patients, and we could start to stain for MCL1. And what you can see consistently across the different patients is clear increase in expression of MCL1 at the protein level at the time of relapse compared to pretreatment. Actually, what you'll see from this plot on the right is that some patients actually had vast increase with relapse. Others already at baseline had already a higher level. And so as we started in our very small cohort, we had 6 patients, we identified 4 others. So we could see that those that had MCL1 high at baseline seemed to have lower survival than those that had MCL1 low at baseline. In terms of AMP-K expression, we looked at AMP-K expression as well as the downstream targets, ACC and phospho-ACC. And again, you can see clearly that in the post-treatment relapse setting, we see higher expression of these protein markers than pre-treatment. And so really this brings me to my last slide, and which is that we're really, I think, through the process of systematically looking at determinants of resistance, we're uncovering novel mechanisms. Certainly, we know that venetoclax is an exquisitely potent and sensitive on-target BCL2 inhibiting agent. But we do understand that MCL1 overexpression is one route to resistance, and also changes in the OXPHOS pathway that can be mediated by deregulated expression of some of the transcriptional regulators is another. And certainly this can help us think about novel therapeutic combinations. I want to thank all the people that have been involved in this. I'm very much beholden to the awesome people in my laboratory, close collaboration with the Getz group. We've had very productive collaborations longstanding with the folks at MD Anderson, and new collaborations with our colleagues in France, and more locally, our local community, both of Dana-Farber and the Broad, have been absolutely instrumental to the work. And so thank you so much for your attention.
Thank you, Kathy, for a wonderful talk. We do have time for a couple of questions.
Arno Kater from Amsterdam. Kate, very nice story, very new data about MCL1. The question is, so in the beginning you showed all these different genes or proteins that were upregulated. You showed NOXA and you showed B-cell XL. And then you started with metabolism, and then it all ended up as MCL1. So you think it's indeed all the others are more or less something on the side. It's really MCL1 that dictates resistance, or you think part of the story is MCL1, but it can still be B-cell XL or BAX/BAC or anything else?
Yeah, so I think the short answer is I think it's complex. So I think MCL1 overexpression is part of the story. I think we've heard from other colleagues that BCL2 mutations are part of the picture. I don't want to exclude that there's other mechanisms, but the kind of the top things that floated to the top for us in the study were MCL1 overexpression, but certainly others can play a role as well.
And then therapeutically, because MCL1 is under control of the TORC mechanism pathway, so if you would inhibit mTOR, do you think you can alleviate actually venetoclax resistance by alleviating the OXPHOS and therefore MCL1 overexpression?
I'm probably the least informed to answer that question, but I'd be happy to talk to you about it later.
Okay.